Thank you, Anna, for the introduction and chairing the session. And thanks, Roy, Sanjun, Stephen, and Roderick, for organizing this session. I'm really happy to have this opportunity to report some of recent studies in my group. I'm talking about a few attempts we have made for denoising a large pharmacogenomic data sets. So let me give a brief introduction on the background first, and also how we got into this field. We started this line of research on pharmacogenomic studies a few years ago. And the typical pharmacogenomic studies involve drugs tested against panels of molecularly characterized cancer cell lines. So the typical data involved will be including drug response measurements, like the drug sensitivity data listed here. So I will explain a little bit more, since that is the major data set we will work on, and also gene expression data, and also other types of molecular profiles of the cancer cell lines. The major goal of pharmacogenomic studies is to connect the drug response data with molecular profiles of cancer cell lines together, to identify meaningful biomarkers, that can be predictive of responses of cancer cells, to individual drugs. So the drug sensitivity data is kind of the data we have been focusing on, and you can see that. So the in the matrix here, so in this particular matrix, so the columns here are drugs, so they can be FDA approved or pre-approved drugs. And the x-axis here are cancer cell lines, and we also have molecular profiles, associated with these cell lines. So the values here, they will be summaries of drug dose response curve. So basically, drug with varied doses will be used to treat, the cancer cell lines, and then there will be a drug response curve produced, and there are different methods to summarize that kind of curve, to tell how effective, the drug is to treat the cancer cell line, or how the cancer cell line is responding, to the particular drug. So there are like AUC, area under the response curve, and there are also IC50 or some kind of other possible measurements. So this is the drug sensitivity data. But for the pharmacogenomics data set, there is a quite long history of data already, and there are a lot of data sets have been generated, during the last, you can see, more than 20 years. And from the beginning, so the scale of the data sets was relatively small. So you can see that just like 59, cell lines. So the drugs can be big, so they can be just a lot of different types of compounds. So as time goes by, so more and more, especially recently, for the data set, you can see that we have relatively more cancer cell lines and more drugs, that have been experimented, together at the same time. So I will first give a brief introduction on a few studies in my group, and then I will focus on the work on denoising the drug sensitivity data set. So one study in my group is about leveraging the molecular data in drug discovery. Actually, that was the topic I was originally planning to present in detail, and then I realized some of the audience have heard this talk already, and multiple times actually. So that's why I decided to change. But I will mention very briefly what this study is about. And so in this study here, so this one is now involving drug discovery data, in the analysis stage. We only use the drug discovery data, for the validation purpose, even though it's not perfect, but it can tell a little bit about the results. So it's trying to connect, diseases with drugs. So what's doing here is that, we have gene expression data like for disease samples and also control samples. And then just by comparison between this disease and the control sample, we can get a list of disease differentially expressed genes. So the genes on the top or on the bottom will be significantly upregulated or downregulated. And then we also have, the gene expression data, from treated samples and untreated samples, for particular drugs. And basically the comparison between these samples will give us a list of genes, that are showing some differential expression under the drug treatment. So you can see that, also the drugs, on the two ends, the genes on the two ends will be the interesting genes because they are kind of the genes showing differential changes, before and after the drug treatment. So the motivation here is that, people believe, so if somehow the drug, differential expression leads the genes, they are having some kind of reversal correlation with this disease differential expression genes, then it could be indicative that this drug, may be a useful, treatment, candidate for that disease. So basically, so drugs that reverse expression of a spectrum of disease genes may be a therapeutic agent. So under this motivation, there have been efforts for finding ways, to try to find this kind of reversal correlation between these differential expression lists. But intuitively, you can see that, we need to focus more on the genes on the two ends, right? So the significantly upregulated, significantly downregulated. So how to select that kind of cutoffs and how to really weight those kind of genes in assessing such kind of reverse correlation. So those are the things we need to consider. And we have developed a method based on kind of statistics, to assess such a reverse relationship. So I will skip the details of the kind of statistics in this talk, but for anyone who's interested, I'm happy to talk about with you, after the talk. So this is one study we did. And another study we did is that we try to have developed some kind of understanding on the cancer cell lines, especially try to do some kind of grouping of the cancer cell lines. So remember that what kind of information we have the cancer cell line. So at least we have the drug sensitivity data, which means that for each cancer cell line, we have the responses, of the cancer cell lines to a list of the drugs. So the number of drugs can vary from, hundreds, to it's from tens to hundreds. And we also have the gene expression, measurements for the cancer cell lines. So if we want to group the cancer cell lines, we want to combine the two data together. So the way for us to group the data together is that for each small cancer cell line group, we try to build this bipartite graph that is showing the gene and the drug relationship. So we saw that, we claim that is the gene drug association pattern. So we build this one through a sparse canonical correlation analysis between the drug sensitivity data and the gene expression data. And then for each small cancer cell groups, we will have this kind of gene drug association patterns to describe it. And then we can group the cancer cell line, small groups into relatively, larger groups based on comparing this kind of patterns or bipartite groups. So by this kind of grouping, so we are able to link some cancer cell lines of different diseases together. And that kind of grouping is more rely on, potential treatment methods, right? So it's based on the common or similar gene drug, association patterns. So this work was published last year in bioinformatics. So that was a really early attempt for us to try to combine the drug sensitivity data and the gene expression data together. But through all of this analysis, we realized that, so the drug sensitivity data is not really that ideal, in terms of quality. So like people realize that even though, we follow, almost the same protocol to do the experiments, and we are working or have the collection of the same cancer cell lines, and we have, the same collection of the drugs, but the drug sensitivity, data matrices produced from different labs can still be quite different. So there is a consistent issue, between data sets. And so we tried to make some comparisons, between data sets from different groups. So some interesting observations were made. So like we found that, the cell-wise correlation. So of course, it's across the same collection of drugs and for the same cancer cell line. So that correlation is stronger compared to the drug-wise correlation. So the drug-wise correlations, if you calculate correlation, the correlation is very low. Even though you consider the size of the vector, try to use some approximation p-values, the p-value, is not significant at all. So, but another interesting observation we observed is that the drug-wise correlation is low. But if we consider the relative relationship about the drug-wise correlations, like we found that if the correlation between drug 1 and drug 2 is high in dataset 1 compared to the correlation between drug 1 and drug 3. So that kind of relationship will be mostly preserved, between the data sets. So basically, like if you just make a very simple, first, a degree of comparison between the data sets, the consistency is bad. But if you just look carefully about some kind of structures of the data, you can see some kind of interesting, relationships, between the data sets. So this means that, there is a hope, for us, try to do something with the data sets to increase, the consistency and to make the data sets more useful in a large scale way. Okay, so with this question in mind, so the first attempt we did is a method. So it's actually in this category, but actually there are a lot of efforts, for people trying to increase the consistency, between the drug sensitivity data sets. Like people are trying to produce or introduce different ways to summarize the drug response curve. And also there are people trying to standardize the experimental protocol, to hope to reduce, some noises, as much as possible, introduced, by different experimental groups. And so the computational effort, that literature is relatively thin at this moment. So a few years ago, one work we did is that we, this is really just a very early attempt, for us to see that just simply by making use of the fact we have observed that the cell-wise correlation is kind of stronger compared to the drug-wise correlation, what we can do. So this one, so basically means that we want to find some operations. So the F here, a G here can be considered as some operations we want to apply on the matrix based on the column-wise, structures of the data or the column-wise, consistency information. And what kind of operations we can find that can help, to improve the correlation, between the rows or between the drugs. So this is not really an objective function because there's no way for us to optimize it. This is really just trying to show that it's kind of the ideal, goal we want to achieve. But what we can really do at this moment is really just to find some reasonable operations, on the matrices, to improve the row-wise correlation. And in that paper, what we did is that we just try to borrow the information between the two data sets, and the things we have observed strong column-wise correlations. So we are using one data set, column as a response, the other data set column as a predictor to do the regression. And we are doing by changing the response and the predictor rows, doing that regression alternatively, to correct and also impute some missing values. And it works very well. But this method still has some kind of weakness. So first is kind of relying on the two data sets, and the two data sets must have a decent overlap regarding the cancer cell lines and also the drugs. But these days, so a lot of studies, they have very focused, diseases, and they have very focused drugs. So we may not be able to find some kind of overlapping, data sets in some way. So we do see a demand to study, to get a method that can be applied on individual, drug sensitivity, data set. And so basically it's just the one data set, not crossing, with other data sets or borrowing information from other data sets. Can we still, try to do some way to reasonably denoise the data and to improve the consistency, between data sets? So if we want to do that, we first need to convince ourselves that there are some kind of meaningful structures in the data that can give us the promise of the hope, to do reasonable dimensionality reduction or do some kind of low rank matrix approximation, to find the to denoise the data. So based on some literature studies, and also our own, exploration of the data set. So we do see, some kind of interesting structures in the data. So like, for example, so there are a lot of horizontal and vertical straps in the data. So those straps, likely correspond to either broadly or narrowly, bioactive, small molecules. For the broadly active molecules, so it means that the drugs are very toxic in some way. So it's kind of killing all of the cancers. And for the narrowly, bioactive small molecules, so which means that they are really friendly, so they are not killing anyone. And for some cancer cell lines, some cancer cell lines are very sensitive to almost all drugs. They just can be killed, very easily. And so basically in our study, we are not really very interested in this kind of drugs or cancer cell lines, right? So it's showing some kind of structure in the data, but that's not the structure of our interest. So we are more interested in, this kind of hotspot. So it's presenting that there is a group of drugs, so they are having some similar, properties, and somehow, they are having similar effectiveness, on a group of cancer cell lines. So there are such kind of groups, available, and also depending on the size of the group. So this kind of structure, can introduce different level of difficulties, for being recognized, in the study. And yeah, also situations like, we have a single drug, and it's an inhibitor. For example, in this example here, it's an inhibitor for some type of mutation, and this drug will make a big difference between cancer cell lines that have or doesn't or do not have that mutation. And so basically for the second and the third kind of signals in the data sets are the signals we want to capture. But there are a lot of more other types of signals, hidden in the data. We don't know yet. But at least, by studying, the drug sensitivity data in this exploratory way, so we know that there is a hope, for some meaningful structures, to be recognized. So the method we consider, is along the line of matrix factorization, because that is providing a way, for dimension reduction and also for the low rank, approximation for the matrix. So with the hope to find a structure, but the traditional, matrix factorization method, when we are using the here, so there are a lot of troubles for us. So like, we don't really know what is the rank, before we do the analysis. So, but for such method, we need to require a specification of the ranks and also the non-convex, nature of the matrix factorization itself, introduce a lot of, challenges for us. So, what we do with this is that we try to explore the use of a version of deep matrix factorization to see that the deep matrix factorization, whether it's kind of helping us to recover some tricky structures that have been missed, by classical or traditional matrix factorization methods. So this is the brief, procedure here, but I just want to emphasize a few things. So the one thing here, so this can be considered a matrix you want to recover. So you are representing it as the product of multiple matrix. So the difference, between the deep matrix factorization and the traditional matrix factorization is about the kind of dimension here. So the original matrix is of dimension n by p, and in traditional matrix factorization method, so the dimension, for M1, M2, W1 or W2 here, they will be less than the minimum of N and P. But in this matrix factorization, we don't have any constraint on what the value of W1 and W2 can be. So basically, W1 and W2 can be larger than NP. Then immediately, this becomes a non-identifiable problem, right? So we have been, introducing, crazily, large search space basically for this kind of deep matrix factorization purpose. But the optimization part can be done through this gradient descent method. And we also introduce residual thresholding, like if there are some kind of outliers, after this kind of low rank, approximation, if those kind of outliers are showing some meaningful particle structures, we want to add them back. Okay, so I want to give some intuitive understanding on this deep matrix factorization. So we are actually ourselves still in the process of understanding the deep matrix factorization better. But basically, we think that compared to traditional matrix factorization, so this deep matrix factorization is making the search space much larger than the traditional one. So the traditional one, to make the problem identifiable, they have to narrow down the search space. And they are trying to find the optimal solution in the restricted search space as much as possible. For this deep matrix factorization here, so the search space is large. It's not an identifiable problem at all. But the optimization procedure or the regularization introduced here is to make the search path is limited. So basically means that we are finding a suboptimal solution in a very large search space. So if the matrix itself is very complicated, having a lot of tricky and hidden structures, so basically this deep matrix factorization method is having a better hope, to recover that. So that is the intuitive understanding about this method here. And of course, just like other deep learning methods, it's very scalable. And also we found that, we don't really have to decide the ranks, of the low rank approximation, and also the result is pretty stable. So okay, so this is showing that how we can decide where to stop. So in deep learning methods, early stopping is very critical, and for us, we are able to check that when there is kind of a big departure between the training and the testing errors, that means that's the part we stop. So if we stop any places around here, we will be doing good. Okay, I don't have much time here, so let me just go to some kind of so simulation results. We make comparison between like robust PCA, and this is our deep matrix factorization, and this is just the traditional matrix factorization, and this is the original structure. So you can see that for some kind of standard structures, so deep matrix factorization is working very similarly with robust PCA. But like for situation like this, when we have two kind of overlapping blocks, so somehow the robust PCA failed, and the matrix factorization, is not powerful at all. And so let me and then for the real data studies, so we worked on these 5 data sets because of their size, and you can see so roughly. So this is one data set, and this is another. So x-axis is representing one data set, y-axis is representing the other data set. And we want to see that the correspondence or the consistency between the data sets. And this is the original one. This is the corrected one. So if you see, the pattern is along the diagonal line, that means the consistency is better. So roughly we can see that after the correction, so it's towarding more, to this kind of a diagonal line. So this shows some level of consistency. And this is a particular example of a single drug. And the purple points is before the deep matrix factorization correction, and the yellow points are after. So you can also see that for individual drugs, between different data sets, after the correction, the correspondence also become better. Okay, so yeah, this is just showing, effects of a drug on this mutated or non-mutated cell lines. It's just after the correction, we can see that we can have a better separation between the mutated and non-mutated cell lines. So these are some kind of example to show the method is working. But as I have mentioned that we are still in the process of exploring the deep matrix factorization for its property and also its application in denoising large data sets. Thank you. Oh, I want to thank my students for their contribution to all of this work. So the deep matrix factorization basically have been done by Tom Hu and Yao Dong, one from Biostat and the other from ECS. Questions?
Yeah, so nice. Yeah, some questions here. Thanks. So it's actually about the first story that you didn't go too deep because you already explained it before, but I didn't know it. So I was wondering, how do you decompose the treatments that the patients could have when you do the comparisons with the cell lines and the different drugs, but patients sometimes already are taking drugs that can be different between patients. So I was wondering how that's.
That one hadn't been incorporated yet, but clearly that's something that will introduce a better resolution of the data. Yeah, so like if we really want to go to the personalized medicine direction, yes, yeah. But the current data set here is, so what we have is really just the cancer cell lines and the drugs. Yeah. So the individual patient effects wasn't really considered. Yeah.
Okay, thanks.
But that's a great point. Yeah, thank you.
So maybe a little bit related to that. Yeah, because the cell lines are like individual entities, and then in a patient we have a more complex tissue. So are you also like working on organoids or something that could resemble better, like the structure of the patient, so that you can also access accessibility of the drug and things like that?
Yeah, if we have such kind of data, and so that's something we want to incorporate. So hopefully that can give us some kind of guidance in terms of the dimensionality reduction. Yeah. So that will be related to when we are introducing or working on the deep matrix factorization method. So whether we can modify the objective function internally to reflect such kind of additional prior information about. Yeah. Thank you.
More questions? Yeah.
I mean, following up on this question about the cell lines and other elements, I was just wondering, one element of the rationale in this matching of transcription signatures, which is what you really ultimately want to achieve, right? Is that the differential expression patterns you find between cases and controls reflect what you would like to revert. And I was wondering whether.
So you mean the first one, right?
Yeah, the motivation slide. So where you really start at the beginning. I was wondering to what extent your model, or the cleaning, also tackles a fundamental challenge here. And one of them is that obviously differential expression between case and control also reflects, many of the consequences downstream of the disease, that are not causative and that you don't want to treat, immune system responses, whatever. Is that something that you're also implicitly hoping to adjust using your deep NMF? Or is it really more technical variation? And if not, is that problem not also something to consider?
So the deep matrix factorization, that method is separate from this, because this one here you can see is really just gene expression data. And for the deep matrix factorization approach I introduced, we are just exploring its application in the drug sensitivity data. So it's different applications in some way. But the points you discussed here, I see that to assess reverse correlation is really just the first step, right? It can just give a very rough match between drug and the disease. But the next step, what other drugs will be really attract, the doctor's, attention, or the pharmaceutical company's attention, to do further experiments. Then we have to do more work further, towards the direction you have just described. Yeah. So.
No, thanks.
Thank you.
Sure, thank you.
To everybody, this we have like 1 hour perhaps. We have correct. Yeah. So we are back at 1, and then we will have a couple of sessions, and then we can go for a hike, right? So we thank all the speakers.